Hello class, today we're talking about Chapter 24, Nutrition, Metabolism, and Energy Balance. So broadly we start off with the concept of nutrients. Nutrients are needed for just regular tasks of the cell, and we broadly break it up into macronutrients and micronutrients depending on how much you need. So macros are things that you need a lot of, the bulk of your ingested food. Carbs, lipids, and proteins. You'll notice that nucleic acids isn't included in the macronutrients. That's because we synthesize our nucleic acids. Water is also technically a nutrient, and we do take in quite a lot of water relative to everything else. Micronutrients are only required in small amounts, and usually it's things like vitamins and minerals that are serving as cofactors or coenzymes for normal enzymatic function. And we'll go into more detail about that. An essential nutrient is one that cannot be synthesized by the body. It has to be supplemented by the diet. And so roughly 45 to 50 different essential nutrients, including some of the amino acids. But we have this idea of use of energy in terms of nutrients quantified by kilocalories or kcals. And so 1 kcal is a dietary calorie. So what you see on a nutrition label is a dietary calorie. And so that is simply a measure of what temperature does it take to raise a kilogram by 1 degree Celsius, kilogram of H2O by 1 degree Celsius. And so it's a measure then of energy. So looking at the vitamins, vitamins are organic. They are organic molecules. They will not have these broadly inorganic characteristics that will be minerals, but they can either be water soluble or fat soluble, lipid soluble. And so water soluble vitamins are processed very quickly. They're processed, and if they're not used immediately, then they're just excreted. The fat soluble vitamins, however, can be sequestered in the fat and actually sequestered to the point that they actually become toxic, perhaps if you have too much of them. So minerals, we're going to separate into our trace and ultra trace minerals. A lot of this is coming through the forms of salts and stuff, especially in our drinking water, but sodium, magnesium, phosphorus, chlorine, potassium, sulfur. These are being used as ions in our extracellular fluids or intracellular fluids. They're being used, of course, to power different reactions. And the ultra trace minerals are somewhat exotic, but they are usually acting as reaction centers for enzymatic reactions. And so probably the best example of these is iron, which is found in the heme protein and serves to basically hold on to oxygen molecules as they move around the body. So starting with our macros, let's talk about the carbs. So carbs can be broken up into complex and simple. Complex is going to be long chains, so starches, celluloses. Simple is going to be mono and disaccharides, so your sort of monomers, your glucose, your fructose, your sucroses. And so you get those through a variety of foods. We call these broadly fibers, depending on how long the molecule is. So an insoluble fiber like cellulose, for instance, we will not be able to digest it completely, so it actually serves as a form of roughage in our digestion. A soluble fiber, however, like pectin, can actually reduce blood cholesterol. So you need both of them. And that's kind of the odd thing out about the carbs, is that there's actually no essential carbs. This is why things like the keto and carnivore diet work. If you need carbs, your body will convert non-carb sources to carbs. So truly there is no essential carbohydrate really, but its use in the body, it is turned into glucose at its basis form in order to synthesize ATP, which is our energy currency. And some cells are using fat for energy, but some are relying entirely on this very simple glucose molecule. And then depending on how excessive your consumption is, you'll get conversion of that glucose to glycogen, which is stored in the liver, or fat, which is stored as triglycerol in the adipose tissues. So 45 to 65% of your total calories should, in a healthy diet, come from carbs. The American diet on the low side with 46% carbs, but it is in the form of these sort of processed complex carbs, usually stuff that is harder to break down. But because of their relative inexpensiveness, they are seen demographically in lower income diets. Moving on to lipids. So the most abundant form, of course, is going to be the sort of solid fats in the form of triglycerides that we get in meat, dairy foods, any kind of oil, hydrogenated oil, and these can come in saturated and unsaturated forms. Saturated fats are more likely to be solid, unsaturated are more likely to be liquid at room temperature. And you also get cholesterol through your diet, although most of our cholesterol is actually synthesized. So cholesterol is not necessarily essential in our diet. And that's going to be found in like egg yolks, the meats, the organ meats, etc. So lipids, lots of purposes. It's adding a layer of protection around our organs. It's adding a layer of insulation because it holds heat very well. And it's our long-term fuel storage. The white of the myelin sheath is actually from the color of the phospholipids that form a layer around it. And the cholesterol molecule itself is actually intercalating plasma membranes, providing structure. Cholesterol ring structures are the precursors of bile salts, and of course steroid hormones like estrogen, testosterone. But also signaling molecules like prostaglandins, stuff that causes pain, for instance. And again, it's just our long-term fuel storage that in heavy use cells is kind of preferred, like the skeletal muscle. It's also a reservoir then for those fat-soluble vitamins. So about 20 to 35% of total caloric intake in a healthy diet. The saturated fats you probably want to limit to 10% or less. And then cholesterol is not actually required in your diet unless you have some other health issues, because it should be synthesized by the liver. Okay, moving on to proteins. Proteins, your dietary sources are split into two depending on how complete they actually are. So it's either a complete protein, something that, for instance, vegetarians really strive to achieve, versus an incomplete protein, which is not going to have all the essential nutrients that will sustain you. And so in this case, I mentioned before that some of the amino acids are actually essential. We do not have the ability to synthesize them. So this is how that works. A complete protein, like animal products and soybeans in this instance, are going to have all the amino acids that you need. So what do we use proteins for? Well, this is really where the rubber meets the road in terms of our biochemistry and metabolism. For instance, structural materials, keratin in the skin, collagen and elastin, the muscle proteins, actin and myosin, of course, and then functional molecules. So biological catalysts like enzymes, and some hormones, for instance, are actually amino acid based. This also has a big role to play in our nitrogen balance because of that amino group that is on all amino acids. And so if you have a positive nitrogen balance, your synthesis will exceed breakdown. If you have a negative nitrogen balance and you're putting a strain on your body, your breakdown is exceeding the synthesis. And so you see this being implicated in whether or not you're using amino acid pools for energy or whether you're using them to actually construct stuff. Because the amino acids actually have like an all or none rule, where if all amino acids are present, then you can functionally build all the proteins that we need. But if something is deficient, those amino acid pools being so transient, now you're going to use them for energy rather than building. So dietary requirements vary pretty wildly because things like metabolism, whether you're in synthesis or breakdown, and your age, because as we age, our dietary needs slowly taper off. A rough rule of thumb, 0.8 grams per kilogram of body weight. And Americans, and especially rich demographics in other countries, do tend to have a sort of meat-heavy diet, so not always an issue for us. Vitamins. Okay. As I mentioned, vitamins are organic compounds. They basically allow for function of an enzyme by serving as a coenzyme. So they bind the enzyme in a very physical way and change its shape so that the enzyme now has an active site that is the correct shape for its substrate. And so these are ingested except for vitamin D, which is made in the skin by the melanocytes, and then B and K, which are synthesized by our microbiome. Beta carotene will get converted into the body to vitamin A. As mentioned, these are water soluble and fat soluble, or fat soluble. Your B's and C's are absorbed with water. B12 absorption, which has, I think, a cobalamin center, it requires some type of intrinsic factor, and these are not stored. Within an hour of ingesting these, you're already excreting them out through the urine. The fat-soluble vitamins would include like A, D, E, and K. They're absorbed with lipid digestion products. They're stored. They're being synthesized in the GI, of course, by our microbiome, except for K. And then if you consume these to excessive levels, they will cause health problems because they're being sequestered in your fat tissue. So what do vitamins do then? Well, they're good in a number of ways. For one, of course, serving as the coenzyme for those enzymes. A great example of that is vitamin C, which is a coenzyme of collagen. If you do not get enough vitamin C, of course, you will get scurvy, which is a problem as we cross the Atlantic over and over without appropriate stores. But these vitamins can also serve as antioxidants, neutralizing the free radicals that are generated during just normal metabolism. And the weird thing is that these are very hard to supplement in some instances to get the same effect. So eating them in a normal, healthy diet is not the same as just taking a vitamin, depending on the vitamin involved. And as we see, broccoli, cauliflower, Brussels sprouts, these sort of brassica type plants are good sources of vitamins A and C, and for whatever reason these are harder to supplement, probably because of like not well understood micronutrients. But moving on to the minerals. Minerals are inorganic in nature. Vitamins are organic, minerals are inorganic. And so 7 minerals are required in moderate amounts: calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium. Typically you'll get these in salt form. And then you have to closely control the homeostasis of these ions because of their various properties in the body. But calcium, phosphorus, and magnesium, of course, used in the bone. Iron, of course, serving as a reaction center for that hemoglobin as it stores oxygen. Iodine serving as the precursor for T3 and T4, your thyroid hormones. Sodium and chloride balancing out positive and negative charges in the ECF and ICF. And sources for this include vegetables, which of course can sequester minerals from the soil, legumes like peas and beans, milk, and some meats, of course. So what is metabolism? We use it colloquially to refer to how well someone is able to partition nutrients and burn fat and stuff like that, but truly it is the sum of all biochemical reactions. And so there are two sides of the coin, right? You're either building molecules or breaking them down, and that is encompassed by anabolism or catabolism. So synthesis versus hydrolysis, turning polymers into monomers and vice versa. So we call this respiration. It's the catabolic breakdown of the food fuels like glucose, for instance, oxidizing glucose to make ATP in the cell. And we'll go over that in more detail in a second. But phosphorylation is a way to take an inorganic phosphate and add it to a molecule basically as energy currency. And this will activate them to perform cellular functions. Another way that you can activate molecules is by passing them an electron. So electrons do have some inherent energy, and so moving them from molecule A to molecule B is the same as adding a phosphate group, essentially. If it's oxidized, it will lose electrons, and with that electron, it will lose energy. Reduced substances gain electrons and energy, and we usually represent this as a hydrogen molecule. And so the two important enzymes act as hydrogen or electron acceptors in this oxidative pathway. Basically, we're taking an oxidized form and reducing it to an energetic form, which will then be sent on to some other pathway and serve as a source of energy. So these redox reactions get catalyzed by some type of enzyme, which usually requires a B vitamin coenzyme, which is why you tend to see the B vitamins pushed for reasons of energy, right? 5-hour energy, what does it have in it? Well, probably a lot of caffeine, but also a lot of B vitamins. And so the two sides of the coin are dehydrogenases and oxidases. The dehydrogenase, as the name implies, is removing a hydrogen. The oxidase is catalyzing the transfer of oxygen. In this case, we would call it an oxidation reaction. And so the molecules that are acting as acceptors in this case are nicotinamide adenine dinucleotide, NAD+, and flavonoadenine dinucleotide. So notably, nucleic acids. Nucleic acids are far more than just information storage and things like RNA, DNA. They're actually being used as energy currency. So moving into cellular respiration, it's not exactly like breathing in and breathing out, which is how we would colloquially use respiration, although the oxygen used therein is important in that. The goal of cellular respiration is to fully oxidize a glucose molecule and from that generate ATP. And so here we have our glucose molecule. We've got 6 oxygens. We are going to turn that into 6 waters, 6 carbon dioxides, 32 ATPs, and there's going to be some heat as a byproduct. There are 3 pathways required of this. Glycolysis, which happens out in the cytoplasm. The citric acid or Krebs cycle, you also see it called TCA, and the ETC, electron transport chain, along with chemiosmosis, we would call this the oxidative phosphorylation. So we're taking our 6-carbon glucose and via glycolysis splitting it into 2 3-carbon pyruvates. We're then taking those 3-carbon pyruvates and turning them into 2-carbon acetyl-CoAs, and the acetyl-CoA is going to interact with a 4-carbon molecule from the citric acid cycle and turn into a citrate or a citric acid molecule, hence the name citric acid cycle. But basically, as this pathway runs, we're going to focus on harvesting the high energy electrons as electron carriers go from oxidized to reduced. We'll also be generating some ATP directly, but we'll get to the details in a second. The electron shuttles, NADH and FADH2, will actually move into the electron transport chain, where they'll power chemiosmosis and the synthesis of ATP. So step 1, glycolysis. As the name implies, we are lysing a glucose molecule. It occurs in the cytosol, actually in the absence of oxygen. And the net energy yield here is only going to be 2 ATP molecules, but also 2 NADH molecules per glucose molecule. So in fermentation, which we'll talk about in a second, you can keep doing this even in the absence of oxygen. The problem is it's just not very efficient, and some organisms prefer it, like yeast, but we're going to want to further reduce the products of this reaction and keep it going. So that was out in the cytoplasm. Basically, we're going to move the products into the mitochondria now. And we're going to take that pyruvate that was split from the glucose, and we're going to push it into the citric acid cycle by modifying it into acetyl-CoA. This will correspond to the release of a CO2 molecule. You can always count the carbon molecules of these products and make some conclusions about what is happening. So a 3-carbon pyruvate split into a 2-carbon acetyl-CoA, and then the other gets lost as CO2. And so CO2 is the most oxidized a carbon can be, so it has lost energy in the process. So pyruvate oxidation yields 1 NADH, so we're generating reduced shuttles for the electron transport chain, and it does it twice per molecule, because remember there are 2 pyruvates. And so now we're going to react those with the products of the citric acid cycle. This will complete the breakdown of that 3-carbon pyruvate to CO2. And so each turn, there'll be 2 turns per glucose molecule, because there were 2 pyruvate molecules, generating 2 ATP, or sometimes an ATP equivalent like GTP, 6 NADH, and 2 FADH2 per glucose molecule. So we did yield some ATP earlier in this step with glycolysis, and we yielded some ATP or ATP equivalents in citric acid cycle, but now we need to take advantage of these reduced electron shuttles by sending them to the electron transport chain. And so there are 2 parts of this oxidative part. The electron transport chain itself is generating then a proton or a hydrogen gradient across that mitochondrial membrane by pumping hydrogen into that space, into the lumen. Then by chemiosmosis, following the gradient down, the energy of the protons will be used to synthesize ATP via ATP synthase. So reduced electron carriers are coming in, they're passing electrons down this electron transport chain, and these complexes are acting as pumps that pump hydrogen from one side to the other. Then the hydrogen will follow the concentration gradient down the ATP synthase. So the electron transport chain itself is the only pathway that actually directly uses oxygen. You have to have oxygen for the other parts of this to run, but it's not using it, so it just has to be there. And so its purpose then is to simply create a hydrogen gradient that can then feed ATP synthase and thus generate ATP. So this is a very reactive process, a very high energy process. And so it wants to do it in kind of a metered way so as not to actually damage the cell with any type of energetic byproduct, and especially the release of heat. So every time the electron is passed, you're going to lose a little bit of energy as heat, but energy is making it through every time, just a little bit less. The problem is these free electrons can actually destroy macromolecules like DNA. So then we move on to chemiosmosis. You have your electrochemical gradient now. The hydrogen is going to want to follow the gradient, but the membrane itself will not let it pass. Right? Hydrogen ions in this case are charged particles. Sometimes it's hard for charged particles to pass across a membrane. So there's only really one way for it to get across, and that is through the mitochondrial membrane channel called ATP synthase. And this ATP synthase does something really clever. There is a small rotary motor that as the hydrogen moves down, it will actually twist and push ADP and inorganic phosphate close together in such a way that ATP now has the activation energy to overcome this sort of synthesis and is synthesized. So glycolysis, you're yielding 2 ATP by substrate level phosphorylation. You're pushing those pyruvate molecules into the mitochondria. So now pyruvate be oxidized to 2-acetyl-CoA. That will generate some reduced electron carriers. Then you'll go through the citric acid cycle. That will yield 2 ATP or ATP equivalents by substrate level phosphorylation. Then again, more reduced shuttle carriers. Those get sent on to oxidative phosphorylation, so electron transport and chemiosmosis, and that will yield another 26 or 28 ATP. So per glucose, with cellular respiration, you're going to yield about 30 or 32 ATP, depending on how you count the initial energy investment phases of glycolysis. And so we use quite a bit of ATP. There were some calculations done that said the average person uses about 500 or 600 liters of oxygen per day. When you do a stoichiometric conversion to ATP from that oxygen, presumably you can make about 120 pounds of ATP per day. and you are using it so fast that, of course, there is an energy deficit even, which is why we can undergo some types of fermentation. And so we would call that anaerobic because we have used up all the oxygen. And now we need to start limiting the damage that is being done. So we will actually shut off some of these molecules and try to regenerate our NADH in order to keep glycolysis going. This is not a long-term energy solution. It only yields about 1/18, so it's actually 18 times lower to ferment the glucose and its products versus trying to push them into cellular respiration. But for us, at least, this is how you end up in lactic buildup. So eventually building up the body's pH. And so looking at some carb reactions, we have some good vocabulary here. Glycolysis, of course, is turning glucose to pyruvic acid. Glycogenesis, polymerizing to form the glycogen, or sort of storage form. Glycogenolysis, which hydrolyzes the glycogen to break down into glucose monomers. And then gluconeogenesis, so forming glucose from non-carbohydrate precursors. So I mentioned before that there's not actually an essential carb. And so this is kind of how you get around that. From lipids and proteins, you can actually build the glucose that you will need for your brain. So our cells cannot store a large amount of ATP all at once. It gets used up quite quickly. And so you need to have a storage form then of glucose in order to feed cellular respiration at a metered rate. And so we do that by turning glucose into glycogen and storing that in the liver and in the muscle cells. Then sometimes when you're in high energy demand, you need to be able to go back to that glycogen and actually peel off glucose molecules. And so there is an enzyme called glycogen phosphorylase that can activate glycogen, especially in response to low blood glucose, and start to send out those monomers to be used as energy. Then you have glyconeogenesis, the process of forming glucose from the non-carb sources. And this is going to be due to a relationship of starvation versus fed states. So in a fed state, you have high dietary glucose, but that will continue to fall quite rapidly. In a fasting state, you will go into glycogenolysis until you are going into early starvation. And then in gluconeogenesis, where now you are starting to cannibalize these non-carb sources to get more of that glucose. So of course there are equivalents of this to the lipids. And because lipids tend to have more carbon bonds, lipids are actually providing a greater yield than either glucose or protein catabolism. So in lipogenesis, you are synthesizing then the storage form of these lipids, so the triglyceride. Cellular ATP and glucose levels are high, so there's no reason for you to use triglycerides as fuel, so you'll begin to store. So 50% will be stored in the adipose tissue, the other 50% deposited in other areas. Now, if you get to a state where you need to use those lipids for fuel, you'll begin to break them down into glycerol and fatty acids. And these will actually be preferred depending on where you are. So the cardiac muscle, resting skeletal muscle, and the liver, like hepatocytes, will use fatty acids rather than glucose as fuel. And so when carb intake is low, you will go into a state of ketosis. And these ketones produced by the liver will be excreted in the urine. This is one of the ways that you can measure whether or not you're in keto. With a little test strip, you can see the actual ketones, and that will tell you whether or not your diet is working. So a good summary figure of how this works. But basically, you're shunting in triglycerides and ketone bodies into various pathways to generate energy molecules. So what about proteins? We mentioned this all or none rule where all amino acids for protein synthesis have to be present, or else the amino acid pool, which is pretty transient, is going to be shunted into other pathways. And so the proteins themselves aren't going to have very high half-lives, not always anyway. and so they need to be broken down and recycled. Well, the amino acids provide a relatively simple building block method of recycling new proteins and different compounds, and so they are not being stored in excess. They are being used for energy or converted into fat. And so the citric acid cycle can turn amino acids that have been processed in such a way into more useful molecules, like for lipids for storage or glucose for immediate use. But first, we have to process it in such a way. And so the process starts with deamination, basically taking that amine group off the protein. And so the body then is using these sort of running pools, trying to balance, how much needs to be here for protein synthesis. If it's not being used for protein synthesis, you can use it for fat storage, or you can use it for glucose storage immediately. These pools are intraconvertible, of course, because they have very common carbon backbone intermediates, and it's very highly organized by the liver, the adipose tissue, and the skeletal muscle. So we're trying to balance then the catabolic and anabolic states in a steady state, in a homeostatic relationship. So the carb and fat pools, easily converted. Of course, they're very closely related in terms of structure, and they can be oxidized directly to produce the energy that our body needs. Amino acids are a little more complicated, of course, because they have one side that's got the amine group, it's kind of basic, and then the other side has a carboxylic acid, so it's a little acidic. So your body's total supply of free amino acids needs to be available for recycling the body proteins, forming the amino acid derivatives that are being used to carry out these various structures, but also the gluconeogenesis, so converting of non-carb sources into carbs in instances where you're going into some sort of ketosis. So we can do that with an absorptive state. This is the fed state that we started off with in that little graph. Lasts for about 4 hours, and this is essentially when your absorption of nutrients is occurring. And so during this time, synthesis will exceed breakdown, and so it's being stored as fats if it's not being used. So for the carbs, of course, your glucose is being used almost immediately, but can be converted if you're in excess. Amino acids, some can be shunted into the citric acid cycle. They can be stored as fat, and most are being used directly for protein synthesis, simply because they're very transient and can't store them. And then the triglycerides, they're very insoluble in water, so it's not like you're moving them around the body, but they can be transported by these chylomicrons. These eventually turn into these sort of lipoprotein molecules that we associate with cholesterol, for instance. A couple different kinds of them, which we're not going into detail about. And so here is the nutrient uptake paths that occur during the absorptive state. Again, this is highly dependent upon the solubility of the molecule in water. And a lot of this is being directed by a push and pull of hormones, especially insulin and glucagon. And so they're basically determining whether something needs to be stored or used. And so moving into the fasting state, the post-absorptive state, the GI tract is now empty. We've already absorbed what we could and we passed it out. The fat gets catabolized, glycogen, proteins will exceed anabolism because we're trying to feed the energy demands of the body. We're trying to maintain our blood glucose until our next meal. And so in this case, we have some sources for blood glucose that include the lysis of glycogen in the liver. This will be your first reserve. Then the lysis of glycogen in the skeletal muscle. And then finally the lipolysis in adipose tissues and liver, and the catabolism of cellular proteins. So during prolonged fasting is when you're going to see cellular proteins used, because this is not an ideal scenario, right? You're starving, your body needs energy, so it's going to begin to break down those proteins. As mentioned, this is controlled by this sort of endocrine mechanism of push and pull, these antagonistic hormones, insulin and glucagon. Insulin is hypoglycemic. Glucagon is hyperglycemic. So if there's too little glucose in the blood, you're going to expect to see a lot of insulin as it tries. It's stimulated by elevated blood levels. It's trying to get diffusion of glucose into the cell. So blood glucose is high, cellular glucose is low. Insulin is going to try and fix that by upregulating the receptors that tell the cell to take the insulin in. Glucagon is the other way. There's too much glucose in the cells, not enough in the blood. So it's rising amino acid levels will also do this. So glucagon is going to promote not only the glycogenolysis, so going to the liver to start to use that glycogen store, but also the gluconeogenesis of turning non-carb sources into carbs. And also lipolysis in the adipose tissue, just trying to get more and more of these glucose sort of precursors released. So glucagon will stimulate a rise in blood glucose levels. So what about cholesterol? Cholesterol is another important lipid molecule. It's not being used as an energy storage source. Instead, it's a sort of precursor. It's a precursor for bile salts, which help us to sort of emulsify fat. It's a precursor for a lot of important lipid-based hormones, and it's actually a precursor for vitamin D. So cholesterol is transported by these lipoproteins. Remember that lipids are insoluble in water. That includes the plasma portion of our blood. And so you actually need some help from a protein to transport these cholesterols and triglycerides. And so there are a variety of types depending on their density. HDLs would be high, LDLs would be low, VLDs would be very low density, and then chylomicrons would be these sort of ultra low that have a micron shape, but almost entirely of triglycerides. And again, these micron shapes kind of form a circle in the as a shape. So here's a good summary of what the various cholesterol types do and their compositions. Probably not going to ask too much about that, but it is interesting to note. So metabolism of cholesterol. So liver is producing 85% of the cholesterol that we need. The rest will come from dieting, and it's such a small amount that even restricting the dietary cholesterol often does not actually influence your blood cholesterol levels. This is very much a genetic condition. Rather, a better way of looking at it is how much is coming from saturated and unsaturated fats. Because saturated fatty acids will stimulate liver synthesis of cholesterol and basically inhibit cholesterol excretion from the body. So saturated fatty acids should make up less than 10% of your diet, whereas the unsaturated fatty acids will enhance excretion of cholesterol into bile salts. And you actually need the bile salts to further basically help lipid digestion. So a trans fat is an instance where a healthy oil is transformed into a solid, perhaps more storable form. And so this is one of the health implications of using margarine for cooking. It has a worse effect on the cholesterol levels than saturated fats. What you see is an increase in the low density cholesterols and a reduction in the high density cholesterols. So how do we regulate this food intake? Well, again, a lot of this is endocrine controlled. It's controlled by hormones, basically hormones making you hungry or making you not hungry. Glucagon is going to stimulate hunger. Remember, it is hyperglycemic. We're trying to get our blood glucose levels higher. Epinephrine is also doing this. Ghrelin, we also call this a hunger hormone, and it will actually peak prior to what you would associate with a typical mealtime. That will change for everyone, of course. And then there are gut hormones being released after a meal to tell you you're not hungry. to tell you to stop eating. So insulin is going to suppress hunger, and then CCK is suppressing hunger as well. Basically telling you we're already about to spend a lot of our energy reserves trying to digest this food. Please stop eating. So in longer term, you have this antagonistic relationship between leptin and ghrelin. And so leptin is actually secreted by the fat cell. And so the fat cells are telling that, hey, there's plenty of long-term storage molecules here, and this is going to protect against weight loss during nutritional deprivation, because it's basically giving you some type of indication how much fuel is in the tank.